MRI of ARL. There is another term called internal auditory betas. It is called IM. There are two pictures of MRI ARL. The first picture is 2D. The second picture is 3D. We can rotate the 3D picture in various angles. Let's start the analysis. First, we need to know the anatomy of our ears. Our ears are outside and inside. Our ears are divided into three parts. The parts are the inner ear, middle ear, and outer ear. Our focus is on our inner ear and middle ear. There is no focus on the outer ear. We know the anatomy of the inner ear. The part that is like an earlobe is called cochlea. The part of the earlobe is called the semicircular canal. There is a part like a earlobe. There is a small bone in the semicircular canal. The step is attached to the bone. The cochlear nerve and vestibular nerve are connected to the cochlea. The back of the step is called incus. The bone is called the malleus. There are three bones in the ear. The smallest bone in the human body. The bones are called the malleus, incus, and vestibular nerve. The earlobe is called the tympanic membrane. The earlobe is called the air drum. The membrane is infected with time. The membrane is broken. The membrane is damaged. There is no problem in the middle ear and outer ear. The earlobe is infected. The earlobe is not heard. There can be various sounds. The earlobe is damaged. The earlobe is damaged. The earlobe is infected. The earlobe is infected. The external acoustic guitar is infected. The temporal bone is infected. There is a muscle under the bone. There is a muscle under the bone. There is fat under the skin. There is fat under the skin. The shape of the outer ear is infected. There are different names for each part of the shape. The upper part of the rocket is helix. The lower part is scapaper and the inner part is triangular dyed part. One helix. And also on the upper part is the outer body. The lower part is canopy of auricular lobe. The ear Thetis MRI can perform two different things. Indication is sensory neural hearing loss. E2 axial 5 mm. This is a brain photogram. We will take 2 brain sequences. The whole brain. E2 coronal 3 mm. We will take a 3D sequence. This is called the space of the brain. It is also called the season or fista of other companies. E1 coronal 2 mm. E1 axial 2 mm. We have 2 axials. One is E2 and the other is E1. We have 2 axials. Post-contrast. If you feel any contrast. If you have any abnormal pathology. If you have any abnormal pathology. Then we will take E1 coronal or E1 axial. Then we will take E1 coronal or E1 axial. If you want to have a radiology. Then E1, post-contrast and E1 axial. Are only required in case of tumor present in internal auditory canal. If there is any tumor. Then E1, post-contrast and E1 axial are needed. We will take 3 images from the localizer. One is sagittal, one is coronal and one is axial. We will take axial plan of brain. We will take axial plan of brain. We know that axial plan is sagittal and coronal. In sagittal, we are paralleling the slices with the corpus callosum. We are paralleling the slices with the corpus callosum. On top of that, from vertex to cervical 1. And to the 1/4 of the corpus callosum. We are playing with that. In coronal, we can see that the slices are parallel to the midline of the brain. The 2 sides of the midline are covered by the same anatomy. We are going to angle the slices. Now, we will take a plan of ARL. We will take a plan of coronal image. We will take a plan of axial and sagittal image. We will take a plan of axial and sagittal image. We are saying that axial plane and angle the planning block parallel to the line along the right and left internal auditory canal. We are saying that we will take the brain. We will take the brain. We will take the brain and see the corpus callosum. When we plan the corpus callosum, we will plan it parallel to the corpus callosum. The black areas are the temporal bones. The size of the temporal bones are covered by the temporal bone. We will plan the sagittal so that it is parallel to the brain stem. The midbrain and brain stem are parallel to the sagittal. Now, we will plan the 3D. 3D planning is an axial protocol. The 3D slices are 0.5 mm. That means there are many small slices and many large slices. That's why 3D sequencing takes time. That's why 3D sequencing takes time. The plan is not like the previous one. The sizes are determined by the internal auditory meters. And the hippocampus, first. The C1 vertebral body, then the C1 vertebral body. That's how we plan the internal auditory meters. From the hippocampus. Now, we will plan the axial planning. We plan the coronal and sagittal corpus. In the previous protocol, we have taken the coronal corpus and planned the sagittal corpus. In the coronal corpus, we plan the slices parallel to the right and left internal auditory meters. We can see the sagittal corpus. We can see the sagittal corpus. We can see the besties. I will draw a picture. The right bestie is the cochlear part. The left bestie is the cochlear part. The nerves in the bestie are connected to the brain stem. This is the semicircular canal. The besties are the cochlear. The nerves are connected to the midbrain. We plan the slices parallel to the right side. We plan the mid-slice parallel to the right side. We cut the cochlear, the besties. And we plan the two sides. And verify the planning drop in the other two plans. We plan the sagittal corpus. The sagittal corpus and the brainstem have the same size. The brainstem is the midbrain. We will give the sizes. We will give the sizes. We have some brain cells. We have some brain cells. We have to see some cells. We have to see some cells. This is the maxillary sinus. And this is the mucus. 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 These are the microas, its pathology, and its secondary sinusites. Then we go to the next step. In the second step, we see the area of the air and the two sides. We have some vestiges here, and here we see a small nerve. We see that the black part in the white fluid is the nerve. Then we have another nerve. This is the corona limace of the air. So this part, the white part, let's mark it. This part is, what will be its name? This is the cochlea. This is the cochlea. Let's see this in MRI. Then our vestiges are here and here. Then the long part of this part is our nerve channel. The brain stem and the vestiges of cochlea are here. The nerves are here from the vestiges of cochlea to the midbrain. And the other part is our temporal bone. If there is an infection in the temporal bone, it is called a mastoid AIDS. Here we see that there is a signal change in the bone, there is a slight fluid. So here is 9. Here is a slight fluid, that means it has a mastoid AIDS. Now these are our 3D images. It is a thin size of 0.5 mm. So if we take 3D images, we should not see the nerves. Here we see the black part. These two black parts are in the middle of the vein. We see that the black part is in the middle of the vein. Here we see that there is a nerve. Here we see that there is a nerve. There are many nerves, so if we take a thin slice, we cannot see the nerves. The reason for taking a thin slice in 3D is to show the nerves. Another thing is that we can make a 3D image. We can make a MIP image. The image below is a 3D image. Or MIP image. This image is the image that we can cut out when we MIP these thin slices. We can cut out the semicircular canal and the veins. We can cut out the other parts of the body. We do not have to cut out the other parts. We can cut out the fluid in the front. We can cut out the other parts of the body. We can rotate these parts in different ways. We can imagine or film these parts. If we want to see the right side, we can cut out this part. We can make a windfall. If we take a 360-degree shot, we can film the photos around you. I will zoom in and out to show you the results. This is the cochlear and nerve area. This is the T1 axial image of the ear. This is the ear area. This is the bone area. This is the nerve. This is the nerve. And this is bone. So normally, our temporal bone or fast-wired bone is black. Because this black bone means there is no pathology or infection in our bone. This black bone here is normal. This is our bone. Temporal bone. Here we have to see another important thing. This is. See this. What is this? This is our midbrain part. And this part is cerebral. So this is midbrain and this is cerebral. Now, this midbrain and this cerebral. The angle here is made. And here is an angle made. This angle is called cerebellopontine angle. So I will show it again. If I don't say midbrain, I will directly say pons. This is pons. This is pons. And this is cerebellum. So this pons and cerebellum's angle is called cerebellopontine angle. So CPA. CPA. Many times doctors have written in advice. MRI of CPA. MRI of air. Special attention to CPA. If we don't know the meaning of CPA, we have to give attention to CPA. CPA is cerebellopontine angle. So pons and cerebellum's angle are often tumor. From this nerve, from the base nerve, tumor occurs. And this tumor is called spinal nerve. So we will see this tumor in detail. This is also coronal image. Like AT2 coronal. These are our. Here are all tissues. Cerebellum's corneal part and nerve. Now we will see the original image of MRI. This is vestibular. This is vestibular. These are round and round. This is semicircular. This is our cochlear nerve. This is cochlear. This is vestibular nerve. This nerve is going to vestibular nerve. And this nerve is going to cochlear nerve. This is cochlear nerve. And this is vestibular nerve. Here is another nerve. This is our mouth. This is facial nerve. If we zoom in more, we can see. This is vestibular. This is cochlear nerve. Here is inferior vestibular nerve. And again there is an apical and middle turn in cochlear. Here is facial nerve. Facial nerve and cochlear nerve are close together. Then superior vestibular nerve. And posterior semicircular nerve. We need to know more about these things. This is the best image. Let's see the nerves. Here we can see the black and black. It looks like a black and black. These are our vestibular corneal. These are nerves. It looks beautiful. The second nerve is again. The second nerve is again. The second nerve is the nerve in the internal artery canal. The nerves are shown. The third nerve is the nerve in the inner artery canal. Here we can see the cochlear and semicircular canal. The blue color is the blood vessel crossing nerves. Blood vessels go through the nerves. This is 3D anatomy. We can see the 3D anatomy in the first process. Here we can see the cochlear, vestibular, and semicircular canal. We can see the right side of the right side. And we can see the left side of the left side. Now we come to the pathology. In the first image. In this part of the ear. In the cerebellum, in the confine angle. We can see an abnormal mass relation. We can see an abnormal mass relation. We can see an abnormal mass relation. When we put the contours. When we put the contours. We can see that the relation is normal. Contours are enhanced. This is the tumor. This is the tumor. This is the tumor. Here we can see another pathology. This is the subphrys muscle. This is the subphrys muscle. Here we can see a little labyrinth. In cochlear, vestibular, and cochlear. In charge of infection. Labyrinth is called labyrinthitis. Labyrinthitis. In the colostrum. In the temporal bone. There is a lot of problem. In the tumor. In the stool. Corsorectal tumor. And the autostress. The autostress in the middle. And in the temporal bone. Infected. Then at the right side. Mastocytitis. Mastocytitis on the right side. Mastocytitis on the right side. Bilateral mastocytitis. Then. And then it comes from lipa, there, small goal. Wishandes is the deaths of the vegetables, their great goal. And then I will end my segment here. Thank you.